All right, this should be review for you. Just a real quick discussion of heredity. We've known about this for a long time. We've done things to benefit us instead of the organisms like these seedless bananas and these giant brown Swiss cows. But we really didn't understand what was going on. So in the 1850s and 60s, this guy studied pea plants and he determined that there were two factors controlling each trait. So Mendel was a monk and they grew peas for their main source of protein. He noticed there were different characteristics. So he started studying those. These factors are now called genes. He didn't call them that. The word gene comes from the same root as generate, to make, to create. So Mendel proposed that one gene came from each parent. And all the traits that Mendel looked at had exactly two versions: yellow seeds or green seeds, or purple flowers versus white flowers. And the only reason that he was able to determine what he did was because he chose traits that had exactly two versions. A lot of human traits have multiple versions. A lot of other organisms have multiple version traits. All of Mendel's had exactly two. And he determined that when they're both there, only one is seen, and that's called the dominant. You need one copy of the dominant factor or gene to make the dominant appearance. But the recessive only shows up if it is the only one there and there's no dominant. The common trait is not necessarily the dominant. For example, in humans, there's a trait that makes 6 fingers on your right hand. That's the dominant, meaning if you have that gene, it shows up, but it's not common. There are many disease genes that are dominant, for example, Huntington's disease, and they're not the most common form of the trait, but dominant means that one copy of the DNA gene and the trait shows up. So what he did was he took true breeding or purebred plants, crossed them. The one that shows up is the dominant, so in this case it's purple flowers. And then he saw that the recessive reappeared in the grandchildren, the F2. So P stands for parent, F1, filial 1 in Latin, first son, filial 2, grandchildren. These different versions are called alleles. The word allele means different. And what we see then, the physical trait, the observable, detectable trait, is called the phenotype. So if an individual is homozygous or purebred for every trait, the babies are going to be like them. Homo means the same. Zygous refers to the first cell that forms the organism, and we'll talk about that in a bit. So, for example, in pea plants, green is recessive, so that means that if the pea plant has green seeds, that is 2 alleles, or homozygous for green. So green pea seeds and green pea seeds make green pea seeds always, because that's the only gene that can be passed on. So the opposite of that is yellow being dominant. So if there are 2 alleles for yellow, that would be called homozygous dominant. 2 alleles for green would be called homozygous recessive. Again, homozygous means both alleles are the same. The genotype is written out using the first letter of the dominant form of the trait. So if we're talking about seed color, we're going to use the letter Y, a big Y for yellow and a little y for green. Hopefully all of this makes a lot of sense to you. And so homozygous dominant, big Y, homozygous recessive, little y. And if they're different, the prefix hetero means different. Heterozygous is big Y, little y, one of each. Hybrid is another way to say that. So we're going to switch gears entirely and talk about cell division again. Meiosis, which literally means the process of reduction, is a cell division that is for making babies. It has 3 distinct differences from mitosis, so hopefully you remember mitosis from a couple weeks ago. There are 2 complete cycles: prophase, metaphase, anaphase, telophase, prophase, metaphase, anaphase, telophase, twice. And so there's no DNA replication in the middle because meiosis is a process of reduction. And so we go from the normal diploid number of chromosomes down to a single set haploid. So the second cycle of division is exactly the same as mitosis, except there's only one set of chromosomes haploid. Haplos means single in Greek. So the second major difference is that chromosomes swap parts, crossing over. The word homologous literally means the same words. So for example, if you have a chromosome that has eye color from your mom, you have a homologous chromosome that has eye color from your dad. And so one homologous chromosome comes from mom, one from dad, and they swap parts, they shuffle the deck. So here we can see the blue representing the paternal chromosome, the pink representing the maternal, and they swap parts. These 2 homologous chromosomes together have 4 chromatids, and we call that a tetrad because the prefix tetra means 4. And this is why you don't look like your siblings. And then the third major difference, the homologous chromosomes separate in anaphase 1 instead of the X-shaped chromosomes coming apart. The sister chromatids stay together in the first round, and then they separate in the second round. So there are some keystone statements. You can look at the slideshow and read those in detail. So the whole purpose of meiosis is to form gametes. Monogamy is what's legal in Pennsylvania. It literally means one marriage. The word gamete means marrying cell. And this makes sense because two of these gametes have to fuse together to form one. And when they fuse, they're called the zygote. That word literally means hitched in Greek. And so the zygote is the two gametes hitched together to make the baby. And the gametes are haploid reproductive cells. So in other words, a single set of chromosomes that would make the zygote have two sets of chromosomes, which is called diploid. So meiosis in lower organisms, that is those without gender like yeast and certain types of pond scum, and males of higher organisms like plants and animals, produce 4 equal gametes. When it's talking about males, those gametes are called sperm, and so the word is spermatogenesis, the process of creating spermatids. And so it looks like this, 4 equal gametes, and this is the same process that yeast or other sexually reproducing, very simple life forms go through to produce haploid gametes. In females, though, the process of oogenesis, literally egg-making process, is not equal gametes because that one large egg cell is where all of the organelles and cytoplasm of the functioning zygote come from. So when you were first you, everything except half of your DNA came from your mother. Your father gave you half your DNA, and the rest of the functional cell came from your mother. So the 3 small genetic trash cans that are produced are called polar bodies. The word polar means to the end. So the DNA is moved to the end, and these are recycled. And so it looks something like this, where we have 1 large egg produced and 3 tiny little genetic trash cans called polar bodies. And so what alleles are going to be in there? Back to the genetic part. So if an individual is homozygous, we don't even have to worry about any kind of probability or Punnett squares or anything like that, because that's the only gene they can give. So if an individual is homozygous dominant for yellow seeds, big Y, the only allele they're ever going to pass on is big Y. Where we have to do some kind of analysis or figure things out is whenever one or both parents are heterozygous. Again, hetero means different, so they have 2 different alleles, big Y, little y. Either one's going to be passed on with equal probability. One of the mistakes that students make sometimes is that, oh, there's a dominant in the parent's genotype, so the dominant will always be passed on. No, it's just like flipping a coin. One or the other will end up in the gametes, and so we need to use some kind of process of modeling this random probability. And we use Punnett squares. Punnett was a British genetic researcher, early 20th century, and he developed this little probability table that we can examine all of the possible outcomes of a cross or a mating. And so each column in the Punnett square represents gamete genotypes from one parent, and the rows represent gamete genotypes from the other parent. And then each box represents possible genotypes of the baby. And so in the example of humans, if you're doing a simple Punnett square and there are 4 boxes in it, you could get 4 genotypes. However, there's only going to be one baby, and it's just random which one the baby actually has. And so consider this example. We have 2 heterozygous yellow-seeded pea plants. Again, hetero means different, so that means big Y, little y. So the ovules could have a big Y or a little y. The pollen could have a big Y or a little y. We put the ovules as the headers of the columns here, and the pollen as the headers of the rows, and we combine those letters, and you can see the 4 possible zygote combinations: 1 homozygous dominant, 1 homozygous recessive, and 2 heterozygous. And so we can actually create ratios of expected genotypes using that. And then phenotype is what it'll grow up to look like. So big Y will be yellow, big Y, little y will be yellow. There are 3 of them that'll be yellow, and little y will be green. And so there'll be 1 that'll be green. And that's a very quick overview of what you should already know about meiosis and Punnett squares. And we'll do some practice this week, and hopefully it'll all make sense. Next week, we'll